Hi friends, welcome to my YouTube channel. This is Arun. Today we will discuss Mannose bending lectin pathway. First introduction. The lectin pathway is one of the three pathways by which the complement system can be activated. The mechanism of the lectin pathway represents one of the first innate immune response to a pathogen. It can be activated through the binding of Mannan binding lectin, L-ficolin, H-ficolin or M-ficolin to carbohydrates one of the pathogen code. The lectin pathway is initiated by the binding of mannose binding lectin, collectin 11 and ficolins to microbial surface of oligosaccharides and acetylated residues respectively. Ficolins are serum complement lectins with a structure of similar to mannose binding lectin and lung surfactant protein SPA and SPD. Ficolins activate the lectin complement system and play important roles in host innate immunity. Then lectin pathway. Lectin pathway is also known as MBL pathway. Lectin pathway activated by binding of mannose binding lectin to surface of microbes bearing mannan in a calcium department manner. The lectin pathway binding causes activation of MASP and cleaves a C2 and C4. Mannose binding lectin associated serine proteases are the enzymatic constituent of the lectin pathway of the complement system. They complement with large pattern recognized molecules such as MBL, other collection and ficolins. Then first step is activated MASP2 cleaves C4 to C4A and C4B. Some C4B binds covalently to microbial surface or pathogen surface. Second step is activated MASP2 also cleaves C2 to C2A and C2B. Third step is C2A binds to surface C4B forming the classical C3 convertase C4B to A, and fourth step is C4B to A binds C3 and cleaves it to C3 and C3B. C3B binds covalently to the microbial surface or pathogen surface. All three pathways there are complement activation, the classical pathway which is triggered directly by pathogen or indirectly by antibody binding to the pathogen surface, the MB lactin pathway and the alternative pathway which is also provides an amplification loop for the other two pathways. And three pathways lead to production of C3b central molecule of complement cascade, and its presence of C3b on surface of microbe marks it is foreign and targets it is for destruction. C3b with two important functions. First one is combines with other complement components to generate C5 convertase, and second one is opsonize bacteria. C4b and C3b are opsonies which are able to bind covalently to glycoproteins on the target cell surface. Opsonization by C3b or C4b leads to engagement of complement receptors on acceptor phagocytic cells, thus targeting foreign particles for phagocytosis. The lectin pathway, like the classical pathway, proceeds through the activation of C3 convertase composed of C4b and C2a. However, instead of relying of antibodies to recognize the microbial threat and it initiate the complement activation process, this pathway uses lectins proteins that recognize specific carbohydrate components primarily found on microbial surface as it is specific receptor molecules. The C3b is said to be acting as an opsonin in this case of because it is enhances the ability of an entity to be phagocytosed. MB lactin pathway is converted into mannose binding lectin binds mannose on pathogen surface, and MBL, MASP1, MASP2 and C4 and C2 is converted into C3 convertase. C3 convertase is divided into two, C3A and C3B. C3A indicates the peptide mediators of inflammation phagocyte requirement, and C3B indicates the binds to complement receptors and phagocytes, and it is opsonization of pathogens and removal of immune complexes. And C3b is indicate the terminal complement components. It includes the C5b, C6, C7, C8, C9, and finally it membrane attack complex lysis of certain pathogens and cells. Mannose bending lectin pathway is initiated by the binding of mannose bending lectin to bacterial surface with mannose containing polysaccharides. Binding of MBL to pathogen results in the association of two serine proteases MASP1 and MASP2. Formation of MBL or MASP1 or MASP2 trimolecular complex result in the activation of the MASPs and cleavage of C4 into C4A and C4B. Complement MASP1 lectin associated serine protease activates coagulation factors and promotes clot formation. However, this is mainly shown in purified or plasma based static systems. Formation of the MBL or MASP1 or MASP2 trimolecular complex result in the activation of the MASPs and subsequent cleavage of C4 into C4A and C4B. The C4B fragment binds to the membrane and the C4A fragment is released into the microenvironment. Activated MASPs also cleave C2 into C2A and C2B. C2 is cleaved into the large C2A and small C2B fragment. C2A binds to the membrane in association with C4B and C2B is released into the microenvironment. The resulting C4B C2A complex is a C3 convertase which cleaves C3 into C3A and C3B. Receptors for C3B and C3BI promote phagocytosis but not the release of toxic oxygen from human phagocytosis. C3b binds to the membrane is association with C4b and C2A and C3A is released into the microenvironment. The resulting C4b, C2A, C3b is a C5 convertase. The generation of C5 convertase is the end of the lectin pathway. Reference: I refer from the given link. Thank you.